The foundational task of genomic research is comparison. To decode evolutionary relationships, biologists must analyze how biological material mutates, shifts, and divides over time. We do this through multiple sequence alignment, a computational method that places the DNA, RNA, or protein structures of different organisms side by side to pinpoint exact genetic variations. When working with a small handful of sequences, this is a mathematically simple task. Traditional algorithms can cross-reference short strings of genetic code almost instantly. But modern genomics does not work with small datasets. Today, researchers frequently need to align hundreds of thousands of highly divergent sequences in a single run. Look what happens to traditional models at this scale. Because older algorithms rely on rigid one-to-one matching, comparing every sequence against each other creates a combinatorial explosion. The calculations multiply exponentially, causing the system to lag and break under the data load. As the sheer volume of genomic data collected globally continues to expand, these traditional methods have become computationally impossible, creating a severe bottleneck at the very beginning of the research process. This bottleneck forces a classic computing dilemma. You can process data quickly by sacrificing accuracy, or you can run rigorous, precise calculations that take months to complete. To bypass this limitation, bioinformatics relies on a specific tool designed to break the paradox, Clustal Omega. It solves the problem without requiring a massive supercomputer. The efficiency comes directly from how the software architecture organizes and parses massive biological datasets. By deconstructing Clustal Omega from the ground up, we can see exactly how its internal pipelines scale sequence alignment to hundreds of thousands of inputs without losing evolutionary fidelity. The software operates as a strict multi-stage structural pipeline. It categorizes the data, calculates the probabilities, and repeatedly refines its own work. Feeding 100,000 raw sequences directly into an alignment algorithm simultaneously guarantees system failure. Phase 1 of the Clustal Omega pipeline solves this through strict organization. This diagram shows how raw sequence data flows into a highly organized structure known as a guide tree. To construct this tree, the pipeline relies on the neighbor joining method. Before doing any deep alignment, this method quickly scans the entire dataset to measure the initial, superficial similarity between sequences. It groups the closest matches together into local branches. The completed tree is essentially a strict set of instructions. It dictates the exact sequential order in which the data will be processed downstream, moving from the most closely related sequences inward. By establishing this queue, the algorithm avoids the computational chaos of trying to compare every piece of data at exactly the same time. The guide tree successfully converts an unmanageable wall of data into a structured, highly efficient, and solvable queue. With the order of operations established, the queued data moves into the primary alignment phase. This phase has to handle a difficult biological reality. Many genomic sequences are highly divergent. They have mutated so far apart that they share very few obvious similarities. To process these divergent inputs, the data enters the HMM engine, which relies on hidden Markov models to execute probabilistic alignment. Traditional alignment tools fail here because they look for exact, physical, one-to-one pattern matches. When sequences have mutated heavily, those direct matches simply do not exist. Hidden Markov models apply a probabilistic framework instead. The engine calculates the mathematical likelihood of an evolutionary relationship between two highly mismatched data points and finds points of probable correlation. Because it evaluates statistical probability rather than exact physical mirrors, Clustal Omega can produce highly reliable alignments even when the input sequences look completely distinct to the naked eye. Replacing brittle pattern matching with flexible, mathematically rigorous probabilities ensures that the system maintains high accuracy across divergent genetic data. Even with powerful statistical models, relying on a single linear pass to process massive datasets leaves a margin for compounding errors. Phase 3 corrects this through an iterative alignment strategy. The output of the HMM engine is routed directly back into its own input. The data is repeatedly fed back through the statistical models. With each pass, the algorithms reevaluate the data, meaning any messy gaps snap into perfectly ordered columns, progressively tightening the accuracy of the entire set. This stability allows for an advanced capability called profile alignment. Once a matrix is locked, new data can be introduced. Researchers can take a brand new, unaligned sequence and match it directly against this locked existing profile. The new data interlocks seamlessly with the solid block. Because the main profile is already mathematically stable, it does not need to recalculate. This bypasses the need to restart the entire alignment process from scratch, saving immense computational time. The iterative feedback loops ensure the initial alignment is perfect, while profile matching ensures that the resulting dataset remains highly updatable as new research emerges. Stepping back from the statistical models, we have to look at how this pipeline actually operates in a live research environment. Phase 4 is the execution layer, driven by heuristic algorithms. Heuristics are specialized problem-solving algorithms designed to approximate solutions incredibly fast. By using heuristics alongside the HHM engine, Clustal Omega drastically reduces the total computing time required for each iterative phase. The software pairs these heuristics with parallel processing, taking the massive calculation workload and distributing it across multiple CPU cores to maximize raw hardware scalability. The researcher controls this entire pipeline through a command line interface. The interface is designed to be highly accessible. It provides a straightforward syntax for beginners to run standard alignments, while exposing deep parameter customization for advanced users who need strict control over the underlying mathematics. It also accepts a wide variety of standard sequence formats natively, easily processing FASTA, Clustal, and Stockholm files without requiring third-party data conversion. Fast heuristics and a flexible interface translate these heavy theoretical models into a highly practical, easily deployable research tool. A capable software architecture only maintains its value if it can adapt to new scientific inquiries. To ensure that adaptability, Clustal Omega is entirely open source. Researchers are completely free to inspect and modify the underlying source code to fit highly specific, non-standard biological inquiries. It is supported by comprehensive documentation, step-by-step video tutorials, and detailed user guides, lowering the barrier for new bioinformatics students. A vibrant network of community forums provides space for vital troubleshooting, peer-driven insights, and shared methodology. Clustal Omega remains the industry standard because this global network of researchers constantly tests, documents, and iterates on its foundation. Unmanageable raw data is structured by guide trees, accurately matched by probabilistic hidden Markov models, and scaled through rapid iterative refinement. This specific pipeline decisively solves the historical computational trade-off between sequence alignment speed and biological accuracy. The deployment of this tool connects directly to the broader human pursuit of mapping the tree of life. By mastering Clustal Omega's architecture, researchers have the capacity to parse the sheer scale of genetic data available today, revealing the hidden evolutionary relationships that define biology.